what I found interesting was your early switch from academia to industry. So I was curious about, reflecting back to that time, what did you find as differences, and what was most challenging as a basic scientist or as a PhD?
Well, I think one of the things people worry about in industry is they'll be told what they have to do. And in my experience, industry is actually crying out for people with a good academic record who know what they want to do. They don't want to be bringing people in and telling them you must do this or you must do that. They want people who have the ideas that they wish to pursue, but perhaps having been committed to that particular course, they have less freedom to go off at a tangent than some academic scientists do, because you have now a very clear objective, and that is to produce a novel drug which will achieve the effects that you aspire to achieve. The other thing, which of course is very, very striking, is in an industrial setting, if you're successful, the last thing you're short of is resources. And indeed, if you start to be genuinely successful, the high probability is you get more resources pushed your way. So if you really have an idea that you'd like to pursue, you think you've got something that could technology that could underpin a new class of drugs, industry is a wonderful place to pursue it. And I felt very comfortable doing what I did. And I have absolutely no regrets about going into an industrial environment. So I spent almost 20 years at SmithKline and French, working closely with medicinal chemists on a number of research programs. And we, in a sense, we were our own masters. The judgment was, did we come up with something worth development through our exercises? Now, one of the things that I was involved with, let's talk about a success, was a dopamine D2 agonist, which is used in which is marketed and was used in the treatment of Parkinson's disease. It's not a cure. I don't deceive myself on that front, but it does help symptoms very, very much. And I've spoken to people who've been very, very satisfied and grateful for what we did on that front. I also know the other side of working in the pharmaceutical industry, when you think you've achieved your objective, but now your compound goes into development, and the toxicologists, the pathologists, or the metabolism people come back and tell you that this compound may be very interesting when you look for its efficacy, but it hasn't got the right characteristics to be a drug. So there are quite a lot of highs and lows working in the pharmaceutical industry.
I gotta ask, because then you got quite heavily involved into patenting.
Yes.
Maybe you can tell us, about the patenting part, and also it was quite interesting on how, getting into patenting and shaping patenting can benefit this type of endeavor, like basic research and product development.
Well, after the pharmaceutical industry, I went to run technology transfer at the Medical Research Council. If ever a man landed on his feet and was fortunate, it was me getting the job at the Medical Research Council. This was now in 1990 and a headhunting exercise. Now, those who can think back to 1990, technology transfer was in its infancy, if possibly prenatal version at that time. And although everybody said they do it better in the States, the truth of the matter was they did it better in 1 or 2 universities in the States and no more than that. So now, why did the MRC get involved at the front end? Well, of course, monoclonal antibodies come from the MRC environment. So the first publication on monoclonal antibodies, which won the Nobel Prize for Cesar Milstein and Georges Kohler, was in 1975. The first monoclonals, therapeutic monoclonal to go on the market was just over 25 years later, Synergis. to prevent respiratory syncytial virus infection in newborn babies. So there was a huge long period. Now, when the monoclonals were first discovered, the technology transfer process, as it was meant to be in the UK, was something called the National Research Development Corporation, was responsible for all technology transfer of science that had been funded by the UK government. There was a nasty gap between researchers and this body, and things like monoclonal antibodies fell down the gap. A fact to which Mrs. Thatcher later took great exception. And so in one way, the MRC, I think Di Rees, who was the fellow Welshman, who was the CEO of the MRC at the time, was a man of some vision, but he was also a man who was being driven by criticism which was coming from Mrs. Thatcher and the UK government. That why should we fund research if you create opportunities with commercial value and potentially high clinical value, but you neglect them? And there's quite a large body of people in the world who have great influence who say, well, why do research when you then succeed and it could be useful in an environment to produce products or services that will serve society well, but you neglect them? Mm-hmm. I am 100% to encourage people to do fundamental research, and I don't think the technology transfer process should ever distort what research is done. But in my opinion, there is either you can see it as an opportunity to see your science used to create useful products and services, or if you don't see it as an opportunity, I think you've actually got an obligation.
You are here because you have been also involved with EMBLEM, and EMBLEM is, a venture company of EMBL that I think wants to do what you have done in your career. So how did you help EMBLEM with your experience?
I was invited here sometime in the mid-1990s. I really don't remember quite so precisely, by Fotis to do a seminar on technology transfer and how we'd been doing it. We were beginning to get credit for doing it quite well. And of course, I had this wonderful support from the top of the MRC, because we were the ones trying to stop the criticism coming from government. I'm not sure I realized just how vital our role was at the time in doing that. I might have been quite frightened if I had. So we were getting credit. And Fotis had taken the view, as he put it to me anyway, that EMBL couldn't be regarding itself as really one of the leading life sciences institutions if it didn't have an effective technology transfer function. And at that point, it didn't have an effective technology transfer function. So over a number of visits, the process was being put in place to create what is now EMBLEM. And I feel very good about the fact that I did have quite a substantial role, and which I think is serving EMBL very well. There's always the opportunity to do better wherever you are, but I do think it's doing a very fine job, and I feel very pleased that I had a role in that.